All right, ready to dive deep. Today, we're all about to become like neuron whisperers.
Sounds intriguing.
We're going to unlock how these tiny brain cells send signals using something called an action potential. It's like, imagine a super intricate, crazy fast Morse code happening in your head right now. And trust me, this isn't just some abstract science class. This is like literally everything your brain does. Thinking, feeling, remembering that super embarrassing thing you did in middle school. It all boils down to these action potentials. So to help us decode these neural messages, what exactly are action potentials?
Good question. Think of an action potential as, a neuron's way of sending a quick, precise electrical signal down its axon. Kind of like a chain of dominoes falling over, one after another.
Okay, I can picture those dominoes, but it's not just a steady electrical current, right? There's a specific pattern to it.
Exactly. It's more like a rapid surge, a drop, then back to the starting point. We call these the phases of the action potential. You've got your rising phase, where the electrical charge inside the neuron rapidly increases. Imagine a roller coaster climbing steeply uphill. That's your rising phase.
Okay, so we're climbing, and then...
Then, bam, you hit the peak, the overshoot, where the charge is highest. This is followed by the falling phase, a rapid decrease in charge, like that roller coaster plummeting back down. And sometimes it even dips a little below its resting state before settling. We call this the undershoot.
So it's like a whole roller coaster ride for these electrical signals. Up, over, down, with a little dip in the stomach at the end. And you're saying this whole wild ride happens in just a few milliseconds. Our brains are constantly buzzing with this electrical chatter, and we don't even know. Now, I know what you're thinking. How do these electrical signals actually happen? What's the machinery behind it all?
It all comes down to these tiny gatekeepers called ion channels.
Ion channels. Okay, tell me more.
Think of ion channels like those revolving doors at fancy hotels. They control what goes in and out of the neuron. But these doors aren't pushed open by people. They're controlled by electricity itself.
Wait, so the electrical charge controls its own WN pathway? That's wild.
Wild and crucial for making the action potential happen. We've got two main players: voltage-gated sodium channels and voltage-gated potassium channels. They're both sensitive to changes in the electrical charge across the neuron's membrane.
So it's like a perfectly timed dance. The electrical charge changes, these channels open and close in response, creating this wave of electrical activity.
Precisely. During that rising phase we talked about, the voltage-gated sodium channels open up, letting a flood of sodium ions rush into the neuron, making the inside more positive.
Okay, so sodium channels open, sodium rushes in, things get more positive. But how do we get back down to that resting state? Don't we need to, like, kick those sodium ions out?
Exactly. As the membrane potential hits its peak, those sodium channels slam shut. And guess who decides to show up to the party? The voltage-gated potassium channels. They've been slowly opening this whole time, and now they're wide open, letting potassium ions flow out of the neuron.
It's like a well-rehearsed relay race.
Huh?
Sodium channels pass the baton to the potassium channels, and the neuron goes back to its resting state, ready to fire off another signal. Amazing. But here's what blows my mind. How did scientists even figure out this whole intricate process? I mean, we're talking about events that last milliseconds, involving molecules we can't even see.
It wasn't easy. Scientists had to get really creative. Early on, they used, believe it or not, giant squid axons. They're almost visible to the naked eye.
Wait, giant squid axons. Now that's a visual I wasn't expecting. They used these jumbo-sized neurons to study something so incredibly small.
Exactly. They developed a technique called the voltage clamp, allowing them to control the electrical charge across the neuron's membrane and measure those tiny ion currents.
That sounds incredibly difficult. These scientists were pioneers.
Absolutely. And their ingenuity led to even more sophisticated techniques, like the patch clamp. This allows us to study the behavior of single ion channels.
Wow, it's like having a microscopic window into the electrical soul of a neuron.
Welcome back, Neuron Whisperers. Before the break, we were unraveling the mysteries of action potentials, those tiny electrical signals that make our brains tick.
It's amazing stuff, isn't it?
We talked about how these signals work, the ion channels controlling the flow. It's mind-blowing stuff. But let's get real. What happens when this elegant system gets disrupted? That's where things get really interesting, and sometimes a little scary. Because even tiny disruptions in these electrical signals can have huge consequences.
It's like those movies where one loose wire can bring down an entire spaceship. What kind of things can mess with our action potentials?
Well, imagine this. You're a sushi chef in Japan, highly trained in the art of preparing fugu. or pufferfish.
Fugu, okay.
Sounds delicious, right?
Yeah.
But here's the catch. Fugu contains a toxin called tetrodotoxin, or TTX, that can paralyze your nerves.
Wait, seriously, people eat something that can paralyze them. That's both terrifying and strangely impressive.
Mm-hmm.
But how does it actually work? Is it messing with the action potentials?
You got it. TTX basically latches onto those voltage-gated sodium channels we talked about.
Yeah.
Those gatekeepers controlling the flow of sodium ions. But instead of letting them do their job, TTX blocks them completely, like putting a giant stop sign in the middle of a neuron highway.
So no sodium flow, no action potential, the signal just gets cut off. No wonder it can paralyze you. Your neurons can't talk to each other anymore.
Exactly. And TTX isn't alone. Remember those red tide warnings you hear about sometimes?
Yeah.
Turns out shellfish contaminated during those events can contain a toxin called saxitoxin,
Oh, wow.
which also targets sodium channels, though in a slightly different way.
So whether it's blocking the channels completely or just messing with their timing, disrupting these ion channels is bad news. It's like throwing a wrench in the gears of your nervous system. You mentioned some toxins actually force the channels open instead of blocking them. What happens then?
Imagine the chaos. Batrachu toxin, found in the skin of certain poison dart frogs, does exactly that. It forces sodium channels to stay open way longer than they should, leading to a massive influx of sodium ions. It's like holding the light switch on permanently. Eventually, the bulb's going to burn out.
Wow, it seems like these ion channels are the nervous system's Achilles heel. It's also amazing how something so tiny can have such a huge impact on our bodies. It really makes you appreciate the delicate balance at play here. Speaking of delicate balances, let's talk about myelin, that fatty substance that acts like insulation around our nerve fibers. It's like the protective coating on an electrical wire, right?
Exactly. Myelin is crucial for speeding up the transmission of those action potentials.
Okay.
Without it, those signals would be sluggish, like trying to send a text message over dial-up.
Right.
Myelin allows those signals to zip along, keeping our reflexes sharp and our brains humming.
So instead of a slow crawl, those action potentials are racing down a high-speed rail.
Mhm.
But how does that actually work? Does the signal just jump the whole distance where the myelin is?
That's where things get even cooler. The myelin sheath isn't continuous. It has these little gaps called nodes of Ranvier.
Nodes of Ranvier sounds like a secret society for neurons. What's so special about these gaps?
These gaps are where the magic happens. The action potential actually jumps from one node to the next, skipping over the myelinated sections. We call this saltatory conduction, and it's incredibly efficient, like taking shortcuts through a maze.
Ah, so that's how myelin speeds things up. Those nodes are like turbo boosts for the action potential. But what happens when this insulation breaks down? That's got to cause some serious problems, right?
Unfortunately, yes. In diseases like multiple sclerosis or MS, the body's own immune system attacks the myelin sheath, disrupting the smooth flow of action potentials.
Oh no, it's like the body's turning against itself. So instead of a smooth highway, those signals are hitting potholes and detours, leading to all sorts of neurological problems.
Precisely. And the symptoms of MS can be incredibly varied, from vision problems to difficulty walking to cognitive impairment, depending on which areas of the brain or spinal cord are affected.
And we're back for the final stretch of our action potential adventure.
The adventure continues.
We've covered so much ground, from those tiny ion channels to the myelin sheath acting like a superhighway for our neural signals.
Right.
But like, just when you think you've got the nervous system figured out, it throws you a curveball. Am I right?
Always. The brain keeps us on our toes.
Right. Okay, so one of the things that blows my mind is how adaptable our neurons are. We've been talking about this like classic model of action potential transmission, but
Not all neurons follow the same playbook.
oh, the different rules for different neurons. What's going on there?
Let's talk about your senses.
Okay,
Sight, smell, touch, all those amazing ways we experience the world.
makes sense.
The neurons responsible for these senses, called sensory neurons, often have a different starting point for their action potentials.
Wait, a different starting point. I thought all action potentials started at that little bump on the neuron,
Yeah, axon hillock.
yes, axon hillock. So what's going on there? Why is it different with sensory neurons?
Think about it. When you touch something hot, you need that pain signal to reach your brain, ASAP. Right.
Yeah.
Immediately.
Sensory neurons have evolved to be super fast, so instead of starting at the axon hillock, the action potential often starts right at the sensory nerve ending, the part that actually detects the stimulus.
So it's like a direct line to the brain, bypassing all the usual traffic.
Exactly. No time wasted.
No wonder we pull our hand away so quickly. Those signals are traveling at like warp speed.
Precisely.
But why the different starting point? Is it like a speed thing?
Efficiency. When it comes to our senses, speed is key. Those neurons need to get that information to the brain ASAP so we can react to our environment in real time.
Right, it's like those emergency override buttons, you know? Sometimes you got to bypass the system and go straight to the source.
Exactly.
So are all sensory neurons like this, or is there even more variation out there?
Oh, nature loves variety. Some sensory neurons still use the axon hillock as their starting point, especially for certain types of information or for fine-tuning the signals. It's a complex dance, but the bottom line is the nervous system has evolved to be adaptable and efficient in processing sensory information.
It's amazing how much is packed into these tiny cells. And it makes you realize that what we've covered today is just the tip of the iceberg, really, when it comes to understanding the brain.
Absolutely.
Yeah.
Every answer leads to new questions. That's what makes neuroscience so exciting. There's so much more to explore.
Well said. And on that note, we're going to wrap up this deep dive into the world of action potentials. We've journeyed into the electrical symphony happening inside your head right now. Hopefully your own neurons are firing after all that.
Thanks for having me.
And for everyone listening, thanks for joining us on this deep dive. Keep those neurons firing, keep asking questions, and most importantly, stay curious.